Hello, this is Professor Tiffany Martin with Medical Microbiology, and we are in Chapter 7, Microbial Genetics. And we just finished some videos on transcription and codons, and now we are moving into translation. So we're still working with gene expression, transcription, and now translation. How do we go from the mRNA to the protein? So we're going to magnify in that into the cell, look inside, and see what's going on in the cell inside the cell. Okay, so we have our DNA, right? Double-stranded, double helix there. We have transcription occurring. You can see that transcription bubble. You can see RNA polymerase moving. It is actively transcribing a strand of mRNA. We know that the beginning of the mRNA strand is the 5' end, so it's still going. And you'll notice that the ribosomes have already started translation. This can occur in prokaryotic cells because there's no nucleus, right? Prokaryotic cells, no nucleus, no barrier. This, where transcription and translation is occurring at the same time, does not happen in eukaryotic cells. So this is something specific to prokaryotic cells, and it's made possible because there's no boundary or barrier or nuclear membrane in between transcription and translation. Ribosomes are in the cytoplasm, but so is the nucleoid region. So all of this is going on in the same place. All right, so as soon as that ribosome binding site becomes available, do you remember that on oh, there it is. There that we have the mRNA strand and it doesn't immediately start getting translated. There are some sequences, some nucleotides in here that tell the ribosome to bind. So as soon as that becomes open and available, a ribosome can bind on this, and you can see that happening there. So we're going to look and see what happens as a ribosome binds and how do we actually get translation from that. We're going to need the ribosomes. We're also going to need some RNA segments, which are called tRNAs. And I think we mentioned this briefly before. We've talked about messenger mRNA, and that's what's going to get translated into a protein. But there are other RNAs that got transcribed, but they function as an RNA. They never get translated into a protein. And one example of those are tRNAs, transfer RNAs, and they make that kind of T shape. So that's what's going on here. And in fact, let's look at those a little further with my whiteboard here. So I've got a tRNA shape, and again, it has that characteristic kind of T shape to it. So I have a tRNA here, and the tRNA's job is to come in and bring the amino acid to the ribosome. So there is a place for the amino acid to bind to the tRNA. So tRNAs is made up of RNA, and then amino acid, this molecular structure that's an amino acid, will come and bind here. Now each tRNA is different. Each tRNA is going to be the one that matches with the codon to determine which amino acid it's going in there. Remember, we got about 2022. There's a tRNA that carries different, every single tRNA carries different amino acids. So let's say this one carries methionine. The way it is shaped, it will only bind to methionine here. And that's based again on the shape and the sequence of nucleotides. All right, there's another thing we need to know about tRNA is that down here in this region, there are going to be 3 ribonucleotides. So again, it's just a whole series of, G, A, A, U, C, through here. But right here, these 3 in this location are actually referred to as the anticodon. And that's because they're the ones that are going to match up with our strand. Here, I'll switch colors. With our strand of mRNA. So my mRNA is coming in, it's going to bind into the ribosome. And remember, mRNA is just, again, a series of ribonucleotides, but they're in groups of 3 because these codons, these groups of 3, is going to determine which amino acid is there. And we know mRNA has, for a start codon, is AUG. So in this mRNA, at some point, there's going to be a start codon. Remember, ribosome binding sites and some other stuff. And then when it gets to an AUG, that's going to be a start codon. Well, this AUG is going to line up with the anticodon, and it's going to use complementary base pairing, and it's going to bind to the anticodon. So if this is an A, the anticodon is going to be a U. This is a tRNA, so it'll be a U. If this is a U, this is going to be an A. If this is a G, it's going to be a C. So this anticodon has a UAC. If it has a UAC here, it only can fit methionine here, and that's how we get that specificity when it comes in and determines where to drop off the methionine. So you can see how that works. So tRNAs are very important in translation. Okay, so let's go back and see what's going to happen when we put it all together. Oops. Okay, so there's my tRNAs there, and they're going to bring in the amino acids, and we have ribosomes. Now, ribosomes, we think of them as just, big globular proteins. They're actually made up of two subunits. There is a large subunit and a small subunit, and they're actually separate in the cytoplasm. They come together on the mRNA. So remember, there's that ribosome binding site. You're going to get the small subunit bind. You're going to get the large subunit bind. You're also going to get the tRNA that encodes for methionine, that start codon and the anticodon that's complementary. That also needs to bind. So to get all of that happening, we're going to call that gets called the initiation complex. I don't have a good picture of it. But to start this process, now I'm just flipping through slides. To start this process, we need the mRNA, we need the tRNA, and then we need the large and small subunits of the ribosome. So there's the small subunit, and then this will be the large subunit that comes together all on that. Now, this is obviously not drawn to scale at all, but you get that thing. All of this needs to come together. mRNA, tRNA with methionine, just the start one, and the large subunit and the small subunit, and we refer to this as the initiation complex. This is what gets translation started, okay? The start codon on the mRNA, and then all of these things binding in their proper spots. So what are their proper spots? How does this all fit together? Let's look at the structure of the ribosome and how that works. Okay, so here's my ribosome, my large subunit, my small subunit. We have our mRNA here. Now, as these bind, they're proteins. They're going to take up space, but the way they fit, there ends up being like three kind of spaces within this fully functional ribosome with these sites. So we have these spaces here. A lot of times they're referred to as windows, and we're going to give them a name. So we have the E site, the P site, and the A site, binding with the large subunit and the small subunit and the mRNA. Now the tRNAs will come in and they fit perfectly in this space. Now, depending on what codon is here, depends on what anticodon can bind. So in this case, we had that UAC, I had my AUG, that got erased, but you know what I mean. The only tRNA that will fit into this space has to be able to match here. The other tRNAs will be close, but not quite. So it will keep them from binding. So these spaces are very particular, and they will only accept that, and that's going to depend on where this mRNA and that codon as to which tRNA can fit. Okay, so I have my E site and P site and A site, so there's actually room for 3 tRNAs to be here. So each one of these spaces has a different kind of function. The A site, this is where the tRNAs enter for the first time. So you can think of A for amino acid. So that's the A site. The P site is where the growing polypeptide chain is going to be attached. So there's a tRNA in the P site, and it's going to have be holding on to the growing polypeptide chain. You'll see how that works in a minute. And then the last site is called an E site, and E stands for exit. So once it moves into this space, the next thing happens is that it leaves the ribosome. Because this mRNA and this ribosome, the ribosome has to move over the mRNA so that we can keep this process going. All right, so let's kind of look at this and how this happens. Now, I do want to point out, so A site stands for amino acid. That's where the tRNAs come in for the first time. P site's growing polypeptide chain. E site's for exit. Now, there is one exception, And that comes back to our friend methionine. The start codon, it will come into the ribosome. Remember, we already defined this as the initiation complex. We need that methionine to come in. It actually fits into the P site. So methionine is the only one that fits into the P site. And then within this ribosome, there will be the A site and the E site. Oh, this is bad. Too bad I can't draw. So within this ribosome, there will be an A site, the P site, and then the E site. And the methionine is the only one that starts in the middle, in that P site, and that's for the initiation complex. So we have an initiation complex. We have the A site empty. I have my mRNA running through here. I have my AUG that's matched up to this, but I've got 3 more nucleotides here. And then I have an open site. Well, that's going to allow the next tRNA to come in and match. And the only one that fits in is going to be complementary to this one. So what was it? CUU, I think, was the next one. So the only tRNA that's going to come in is going to be GAA, right? So the one that has that anticodon is going to come in, and that's going to be pulling that amino acid that goes in there, and I've forgotten what that was. Okay, so let's go back to the PowerPoint. All right, so we've got our initiation complex. We have our AUG in this P site, and then let's see what happens. All right, so small subunit, large subunit, there's my mRNA. The ribosomes have bound in that ribosome binding site. I have my tRNA in that middle site carrying methionine. And then my A site was open, and I have my next one come in. Now this is a different thing, so it's got a different amino acid here. This one is UUU for the codon, so the anticodon was AAA. The tRNA that has the anticodon AAA fits in, and that one carries phenylalanine. So now I've got 2 tRNAs into this process. So now what happens? We're trying to build a protein, right? We need to link amino acids. So we've got to link up methionine and phenylalanine. And there's some energy involved with this. There's going to end up being some energy in to make translation happen. But we're going to link up methionine and phenylalanine. So methionine links up with phenylalanine, and now it is now joined to this tRNA because phenylalanine was still attached to this tRNA. Methionine now is attached to phenylalanine. You can see it's no longer attached to that first one. Here, everything kind of shifts over. Whoops. Everything kind of shifts over, and you can see the tRNA that originally brought in methionine is now in the E exit window. The tRNA that is now got the growing polypeptide chain has moved into the P polypeptide window, and my A window is now open. So the ribosome has moved across the mRNA, and now I have an A window open. What's going to happen now? Well, whatever fits with this codon, the tRNA with that anticodon is going to come in, bringing its amino acid. The original one left. It doesn't have methionine anymore. It has left. This one comes in. We get that reaction, and we're now going to join threonine. And my growing polypeptide chain is here. You can see how that shifts over. The growing polypeptide chain is in that middle window. The E is one leaving. The A is the amino acid coming in. Now you'll notice the one leaving no longer has its amino acid, right? It was added to the chain. This tRNA can be used again. It's going to go back out into the cytoplasm, and when it finds another amino acid that fits, then it's going to be charged again, and it'll have that amino acid, and then it can participate in translation again. So that's it. Go through that. I will post some animations. I actually think this site isn't active anymore, but I will post some Pearson animations as well. Let me point out one other thing before we leave this. So we saw this figure, and we saw that translation can start even before transcription finishes. We also can see that translation As soon as that ribosome moves off of that ribosome binding site and passes the AUG, as you saw before, this is moving down the line, this AUG in this ribosome binding site is open again, which means another ribosome, another small unit, subunit and large subunit can combine there and start translation. So you can get multiple ribosomes. This is called a polyribosomal complex. You can get multiple ribosomes translating the same mRNA. And you can see that green growing polypeptide chain, and as the ribosomes get further, the polypeptide chain gets longer. What's it looking for to stop? Where does translation stop? The stop codon, that's right. So it will continue until translation stops. We know this happens because we can see it on the picture. This is kind of a beads on a string, and you can see the black is the ribosomes. This next picture, I like this one better. You can see the black is the ribosomes, and this is the polypeptide chain, and then the mRNA strand is there. So you can see this happening. Okay, that's it for translation. So make sure you get that down, and then we're going to do operons, and then we'll be done with the chapter, I think. So, all right, keep with me.